public health surveillance networks are currently monitoring reports regarding a newly identified strain of hantavirus, which preliminary data suggests may carry a significantly higher mortality rate than previously documented variants. While hantaviruses are a known quantity in the world of infectious disease, any shift in the virulence or genetic makeup of these pathogens necessitates immediate scrutiny from the global scientific community. Hantaviruses are a family of viruses, spread mainly by rodents, and can cause varied disease syndromes in people worldwide. In the Americas, these viruses typically manifest as hantavirus pulmonary syndrome, or HPS, a severe and sometimes fatal respiratory disease. In Europe and Asia, they more commonly present as hemorrhagic fever with Raynaud syndrome, or HFRS. The distinction between this new strain and common seasonal illnesses like influenza is a critical point of focus for health officials. While the flu affects millions annually with a relatively low case fatality rate, hantaviruses are characterized by their rarity and their high lethality. If reports of a new record-high mortality rate are confirmed, it would place this strain among the most dangerous zoonotic threats currently under observation. Understanding why viewers should care begins with the nature of the virus itself. Unlike many common viruses, hantavirus is not typically spread through human-to-human contact, but through the inhalation of aerosolized particles from the waste of infected rodents. This means that environmental exposure in specific settings, ranging from rural agricultural areas to suburban storage spaces, remains the primary driver of infection. The alarm raised by recent data points to a potential shift in how the virus interacts with the human immune system, possibly leading to more rapid onset of symptoms or a more aggressive attack on the pulmonary or renal systems. As we frame this development, it is important to remember that hantavirus is not a new discovery. It was first brought to prominent global attention during the Korean War and again in the early 1990s in the Four Corners region of the United States. However, the emergence of a strain that claims to be deadlier than any previous iteration suggests a biological evolution that requires specialized laboratory analysis. Public health agencies use this data to determine the level of threat to the general population and to issue specific travel or safety advisories if clusters are identified. At this stage, the focus remains on verification and the establishment of a clear geographical and chronological footprint for these latest cases. Officials emphasize that while the term deadlier than any flu is a stark comparison, it serves to highlight the clinical severity of the infection rather than its rate of spread. The primary concern for editors and health correspondents is the lack of early-stage diagnostic clarity, as the initial symptoms often mirror less severe viral infections, leading to delays in the life-saving supportive care that is required for survival. The core developments surrounding this new hantavirus strain are currently centered in high-level biosafety laboratories where researchers are working to sequence the viral genome. When a new strain is reported with record-high mortality, the first priority for agencies like the Centers for Disease Control and Prevention and the World Health Organization is to confirm the laboratory findings. This involves looking at the specific proteins the virus uses to enter human cells and determining if any mutations have occurred that make it more efficient at bypassing the immune system. In clinical settings, hospitals in the affected regions are being alerted to watch for patients presenting with sudden onset of high fever, muscle aches, and extreme fatigue, followed rapidly by acute respiratory distress or unexplained kidney failure. The diagnostic process is complex. Because hantavirus is often rare, many standard clinics may not immediately test for it, preferring to rule out more common pathogens first. However, the record-high mortality rate mentioned in recent reports suggests that the window for effective intervention is narrowing. In the lab, testing for hantavirus typically involves looking for the presence of IgM antibodies, which indicate a recent infection, or using polymerase chain reaction, or PCR, to detect the viral RNA itself. These tests must often be conducted in specialized facilities, which can create a logistical lag between the onset of a cluster and the official confirmation of the strain's characteristics. There is also the question of the rodent reservoir. Different hantaviruses are carried by different species, such as the deer mouse, the white-footed mouse, or the cotton rat. Part of the current investigation involves trapping and testing local rodent populations to see if the virus is becoming more prevalent in its natural hosts or if it has jumped to a new species altogether. Regarding trade and logistics, while hantavirus is not known to be a significant threat to global shipping in the same way as insect-borne diseases, health officials do monitor warehouses and shipping hubs where rodent infestations could potentially expose workers to contaminated materials. At this time, there are no official reports of travel restrictions, but public health guidance remains focused on individuals who work in construction, agriculture, or forest management, where exposure risk is highest. The discrepancy between sensationalized claims and verified data is a recurring challenge in these reports. While a headline may scream that a virus is deadlier than the flu, the verified information focuses on the case fatality rate among those already infected, which historically sits around 35 to 40% for pulmonary strains. If the new strain exceeds these numbers, it represents a significant medical emergency for those in high-risk environments. Scientists are currently contrasting these new claims against historical data to see if the increased mortality is due to the virus itself or due to factors like late diagnosis or overwhelmed local healthcare infrastructure. Prevention and surveillance remain the primary tools in the public health arsenal when dealing with any hantavirus, especially one with a reported increase in mortality. Because there is currently no specific vaccine or antiviral treatment approved for widespread use against hantavirus in many regions, the focus is entirely on risk mitigation and environmental control. Guidance from health authorities is consistent. The most effective way to prevent infection is to eliminate rodent habitats in and around human dwellings. This includes sealing even the smallest gaps in buildings, keeping food in rodent-proof containers, and using traps. However, the most critical advice concerns the cleaning of areas where rodents have been present. Experts warn never to sweep or vacuum rodent droppings, as this can kick up the very aerosolized particles that lead to infection. Instead, the recommended procedure involves wearing gloves and using a disinfectant, or a mixture of bleach and water, to thoroughly soak the area before cleaning it up. Surveillance programs are also being ramped up in response to the reports of a new strain. This involves sentinel programs, where wildlife biologists track rodent population densities and infection rates in the wild. Often, a spike in human hantavirus cases is preceded by an unusually high rodent population, sometimes linked to environmental factors like a mast year, where an abundance of seeds or acorns leads to a population boom. Understanding these ecological triggers allows health departments to issue warnings before the first human case even appears. Despite the alarming nature of the reports, there are honest limits to our current knowledge. It remains unknown exactly why some individuals develop the most severe forms of the disease, while others might have a more moderate response, though genetic factors and the viral load at the time of exposure are suspected to play roles. The public is urged to remain calm but vigilant. The high mortality rate is a serious clinical concern, but the virus remains difficult to catch for the average person who does not have direct contact with rodent-infested environments. Medical professionals emphasize that if you have been in a space where rodents were present and you begin to experience flu-like symptoms, it is vital to inform your doctor of that specific exposure immediately. Early aggressive supportive care, often involving oxygen therapy or mechanical ventilation, remains the best chance for survival. As the situation evolves and more laboratory data becomes available, the global health community will continue to refine its guidance. For now, the focus is on breaking the chain of transmission between the animal reservoir and the human population. For more updates on emerging public health threats and official surveillance reports, consider subscribing to this channel. The core developments surrounding the reported emergence of this new hantavirus strain are currently concentrated within high-containment laboratory environments and specialized infectious disease units. When a pathogen is flagged for record-high mortality rates, the global public health apparatus initiates a multi-tiered diagnostic and investigative protocol. At the primary level, Biosafety Level 4 laboratories, the highest level of biocontainment, are tasked with isolating the viral genome to determine exactly how this strain differs from established lineages, like the Sin-Nombre virus in North America or the Hantavirus in Saint Asia. Scientists are specifically looking for mutations in the viral RNA segments, particularly the small, medium, and large segments that encode the nucleocapsid protein, surface glycoproteins, and the viral polymerase. Any structural change in the surface glycoproteins, known as GN and GC, could potentially explain an increased efficiency in how the virus attaches to human endothelial cells, leading to the rapid vascular leakage and pulmonary edema that characterize the most lethal cases. In clinical settings, the current challenge involves the early differentiation of hantavirus from more common respiratory illnesses. While the source material highlights a mortality rate than any influenza strain, medical officials note that the clinical progression is vastly different. Hospitals are being advised to monitor for a specific prodromal phase, which typically lasts 3 to 5 days and includes high fever, severe muscle aches, particularly in the large muscle groups of the back and thighs, and gastrointestinal distress. The transition from these general symptoms to the cardiopulmonary phase can occur in a matter of hours, marked by a sudden drop in blood pressure and the accumulation of fluid in the lungs. To combat this, labs are utilizing enzyme-linked immunosorbent assays, or ELISA, to detect IgM antibodies, which serve as a marker for acute infection. However, because these antibodies may not be present in the very earliest hours of the illness, real-time polymerase chain reaction, or RT-PCR, is being employed to detect viral genetic material directly from blood or tissue samples. This laboratory data is critical for agencies to determine if the high mortality rate is a result of increased viral virulence or a consequence of the virus spreading into populations with no prior immunity. Simultaneously, field researchers and wildlife biologists are conducting environmental surveillance to identify the specific rodent reservoir associated with this strain. In the history of hantavirus reporting, each unique strain is typically linked to a specific host species, such as the deer mouse, the white-footed mouse, or the bank vole. If the virus has jumped to a new host, or if the existing host population is experiencing a surge in viral shedding, it changes the risk profile for human exposure. International health agencies are also scrutinizing trade and shipping routes, particularly in regions where bulk grain storage or large-scale shipping containers may provide a habitat for infected rodents. While there are currently no verified reports of the virus surviving for extended periods on dry surfaces or non-biological cargo, the risk of aerosolization during the handling of contaminated materials remains a point of high-level concern for occupational health regulators. The contrast between the sensational nature of the deadlier-than-flu claim and the verified clinical data remains a focal point for investigators. While influenza has a significantly higher transmission rate and infects millions, the record high mortality mentioned in these reports refers to the case fatality rate, the percentage of people who die once they have already contracted the virus. Historical data for hantavirus pulmonary syndrome often shows a fatality rate between 30 and 40%. Any elevation beyond these figures would represent a historic shift in the pathogen's lethality, necessitating a total reassessment of global treatment protocols and emergency response planning. The prevention of hantavirus infection, especially when dealing with a strain reported to have record high mortality, relies almost entirely on rigorous environmental management and the disruption of the transmission chain between rodents and humans. Public health guidance remains steadfast in its primary recommendation: the elimination of rodent habitats in and around human structures. However, the most critical practical takeaway for the public concerns the specific mechanics of cleaning areas where rodents have been present. Because the virus is transmitted through aerosolized particles found in dried rodent urine, droppings, and saliva, standard cleaning methods like sweeping or vacuuming are strictly discouraged. These actions can launch the virus into the air, where it can be easily inhaled by anyone in the vicinity. Instead, Official protocols dictate that any contaminated area should be thoroughly wetted with a disinfectant or a mixture of 1 part bleach to 9 parts water. This wet cleaning method ensures that viral particles remain trapped in the liquid and are neutralized before they can become airborne. For those working in high-risk environments, such as cleaning out long-dormant sheds, cabins, or storage units, the use of personal protective equipment, including non-porous gloves and N95 respirators, is highly recommended by occupational safety agencies. Surveillance also plays a proactive role in preventing outbreaks before they reach a critical mass. This involves sentinel programs where wildlife biologists and public health officials monitor rodent population densities and the prevalence of the virus within those populations. Environmental factors, such as unusually heavy rainfall or a surplus of natural food sources like seeds and nuts, can lead to a population explosion among rodent reservoirs, a phenomenon that often precedes a spike in human cases. By tracking these ecological shifts, health departments can issue localized warnings and heighten clinical awareness in specific geographic corridors. Despite these robust surveillance efforts, there remain significant limits to current medical knowledge regarding this new strain. One of the most challenging aspects of hantavirus is the incubation period, which can range from 1 to 8 weeks, making it difficult for patients to recall the exact moment or location of exposure. Furthermore, because the initial symptoms so closely mimic more common viral infections, there is often a delay in seeking the specialized intensive care required to manage the rapid onset of pulmonary or renal failure. While the source reports a mortality rate deadlier than any flu, it is important to contextualize this for the general public. While the virus is exceptionally lethal to the individual, it does not currently possess the high transmissibility of respiratory viruses like influenza or the common cold. The statistic remains largely confined to those with direct environmental exposure to rodent waste. Ethically, the role of public health reporting is to provide an honest assessment of the threat without inciting undue panic. The record-high mortality rate is a serious indicator for the medical community to prepare for more aggressive clinical interventions. But for the average citizen, the risk remains low if proper hygiene and rodent-proofing measures are maintained. As researchers continue to study this new variant to determine if it requires new diagnostic tools or treatment strategies, the best defense remains a combination of vigilance and informed prevention. For more in-depth reporting on global health security and emerging viral threats, consider subscribing to our channel.